everyone, the ITLS Academy. Today we are going to discuss the topic that is your organoleptic and physical parameters. Okay, so what is this organoleptic parameters? Okay, so in today's lecture we are going to discuss about this organoleptical parameters, and in this the first parameter that we are going to discuss is about the color. So in today's lecture, the first property that we discuss is about the color. But first of all, what is this organoleptic properties or physical parameters are? Okay, see, organoleptic properties are used to measure the quality of the food, water, and other substances, and the base that we choose for measuring the quality of all these things is the senses. Okay, and this senses is either taste, smell, and touch. Okay, behalf of all these things we measure the quality of the product, quality of the product, and then after that we decided the shelf life of that product, shelf life of that product. Okay, so there are different organoleptics and the physical parameters are there which decided the quality of the product. But the first parameter that we are going to discuss is about the color of the food product. Sorry, color of the water, not the food product, color of the water that we are going to discuss in today's lecture. So let's start. The first thing that we are going to discuss is that the color. Okay, so color. We all know that this color is what? Color is the first thing that attract the consumer. First thing that attract the consumer. Okay, if it is the point of water, food, if it is the point of water, or if it is the point of other substances. But if the color of the product is not in a good way, then it will not attract the consumer. Consumer means that who consume that particular product. Consumer means that who consume that particular product. Okay, so the color of the water, we all know that it's what? It is a colorless, means that transparent. Okay, if it is not colorless, then do you drink this water, that water? Suppose that this is a water. Okay, now suppose that some pollutant are mixed into that water. Pollutant are mixed with the water. When the pollutant are mixed with the water, it will change the color of the water. Now, can you drink that water? Your answer is no, because the color of the water is change. Okay, the first thing you see when someone give you water for the drink, then first thing that you notice is what? The color of that water. If it is not in a proper condition, then it will not acceptable. So in today's lecture we are going to see that how we measure the color of the water, measure the color of the water by different method. Okay, first method that we are going to discuss in today's lecture. Okay, so first of all, before discussing the method of the color measurement, we are going to see the introduction part of that color in water. Okay, so we all know that color of the water is due to the presence of the inorganic ions. Okay, color of the water is due to the presence of this inorganic ions. Now, what is this inorganic ions? The ions like iron, manganese, humus, pet materials, plankton, weeds, and industrial waste. Okay, all these things decided the color of the water. Okay, so the term that is color is used to mean that true color, that is the color of the water from which turbidity has been removed. Okay, when the turbidity has removed, then the true color of the water is appear. Okay, the term apparent color include not only the color due to the substances in a solution but also due to the suspended matter. Okay, so there are two type of color. There are the color are defined by a two term. One that I already discussed is the true color. Okay, which means that when we remove the turbidity, then the color of the water is called a true color. Okay, now the next is that the next term is apparent color. Okay, now the apparent color means that the color of the substances appear due to the substances present in a solution, substances present in a solution, and along with it the suspended matter, due to which the dark color that appear is called a apparent color. Now, apparent color is also determined on the original sample without filtration or centrifugation. Okay, so without filtration and the centrifugation, the color that we get that the color that we get is called a original sample. Okay, now the next is your this is the method that we use for determining the color of the water. Used to determine the color of the water. So the first method that we are going to use is a platinum and cobalt method. Platinum and cobalt method. So in today's lecture, we are discuss about the method that we use for determining the color of the water is the platinum and cobalt method. Okay, so first of all, we are going to discuss about the principle of that method. We all know that this color is measured by a visual comparison of a sample with a platinum and cobalt standard. Okay, so we take a platinum and cobalt standard and compare a color of the water with it, and it is measured by a visual comparison. Okay, suppose that this is our sample. Now, this is our sample and this is cobalt standard and this is platinum standard. Platinum standard and this is your cobalt standard. Now what we have to we compare this compare the color of the sample with the cobalt standard and with the platinum standard, and via this we measure the color. 1 unit of color or hazen is that produced by 1 milligram of platinum per liter in the form of chloroplatinate ion. Okay, so if we define the 1 unit of the color, then 1 unit of the color or hazen is defined as that produced by 1 milligram of platinum per liter. Okay, we take a 1 milligram of platinum and dissolve into a 1 liter of water in the form of chloroplatinum ion. Okay. Now the next is your interferences. Okay. So there are three interferences. The first one is that slight amount of turbidity will interfere with the determination. Okay. When we measure the color of the water, then slight amount of this turbidity will interfere. Therefore, samples show our visible turbidity should be clarified by our centrifugation. Okay, so this turbidity can be clarified by using a centrifugation. Okay, now the next interference that this method, that is your platinum and cobalt method, are depend upon a pH. Color of water are normally increases with increase in pH value unless the color ion is precipitate. Okay, so the color of the water is increases when the pH value are increases unless the color ion are precipitate. The third interference is that we use a filter paper which may result in the removal of the some color. Okay, due to the use of the filter paper, maybe some amount of the color are removed, which leads to the erroneous result. Therefore, filter paper should not be used for the determination of the true color. So when we have to determine the true color, then in that case, the filter paper should not be used. We have to avoid the use of that use of the filter paper. Okay, so here there are three interference. First one is your turbidity may interfere the determination. The next is your that it is depend upon the pH. Okay, if the pH is increases, then the color of the water will also increases unless there is a colored ion or precipitate. And the last one is that we uses the filter paper, which leads to the removal of the some color particles. Okay, so in case of the true color, we are not going to use the filter paper. Now the next is your sample handling and preservation. Representative sample shall be taken in a clean glassware. Okay, we all know that when we have to perform our experiment, when we have to perform the experiment, now in that case, we have to take a clean glassware. There is a reason for taking this clean glassware, because if the glassware is not clean, then it will interfere in the result. That's why we have to take a clean glass. Now, color should be determined as early as possible after the collection of the sample. Okay, when we collect a sample, as soon as possible, we have to determine the color. Otherwise, there is a biological activity and the physical changes are occur when we store the sample, and it will affect the color of the water. Refrigeration of the water sample at 400 degrees Celsius is recommended. Okay, so when we have to store the water sample for after some time use, then we have to store it at a 4 degree Celsius. But the first thing that is recommended is that we have to measure the color as soon as we take in a sample. Otherwise, there is a physical changes are occur which may affect the color of the sample. Now the next is your apparatus that we use. Okay, so here only two apparatus are used. One is your Nessel cylinder, and the capacity of this Nessel cylinder is around 50 ml. And the another one is your centrifuge or a filter assemble. See, this Nessel cylinder is what? It is a glassware. Okay, and this centrifuge is what? It is a instrument. Okay, so centrifuge or a filter assemble with a glass fiber filter or a membrane filter with a functional pore size approximately 0.45 micrometers are used. Okay, so here we use two things. One is your glassware and one is your instrument. Now the next one is your reagent. Okay, so there are some reagent that we also use. The first reagent is that we have to prepare a standard chloroplatin solution. This is your first one. So for this, what we have to do, we take a 1.246 gram of potassium chloroplatinate. Okay, suppose that this is your beaker. Now inside the beaker, first of all, we are going to take a 1.246 gram of potassium chloroplatinate, that is K2PTC60. Okay, and this is equivalent to this 1 gram of potassium chloroplatinate is equal to the 500 gram of metallic platinum. Now after that, we are going to take a 1 gram of crystalline cobaltous solution. 1 gram, just a minute. 1 gram of crystalline cobaltous chloride solution, and this is equivalent to the 25 milligram of metallic cobalt. Now we taking a distilled water 100 ml. These all things are dissolved into a 100 ml of distilled water, that is your 1 gram of crystalline cobalt chloride or 1.246 gram of potassium chloroplatinate. Now after dissolution, we added a HCl. Okay, this distilled water will also contain a HCl and concentrated form of HCl. After that, again we added a 1000 ml of distilled water into it. 1000 ml of distilled water. Okay, then this standard solution are equivalent to our 500 color unit. After that, when we added our 1000 of this distilled water, then our standard chloroplatinate solution are ready, and it is equivalent to our 500 color unit. Okay, so the first reagent are ready, that is your standard chloroplatinate solution. Now the next reagent is your preparation of the standards. Okay, after that we are going to prepare our standard solution, and the standard solution are in the unit of 5, 10, 15, 20, 25, 35, 30, 35, 40, 45, 50, 60 and 70. Okay, and these all things are diluted by this 5 are diluted by a 0.5 ml of standard chloroplatinate solution. Okay, 1.0, 1.5, 2.0, 2.5, 3.0, 3.54, 4.55, 6, 6, 7. And along with it, we are also going to add a distilled water of 550 ml. Use distilled water as a 0 unit standard solution. Now the next standard is that we have to protect our standard against a evaporation and a contamination. Okay, so when we prepare a standard solution, so we have to take care that the solution should not evaporate or contaminate by using a inert stopper. The standard should also be protected against a absorption of the ammonia, which causes the increase in color. Okay, So we also protect there is a absorption of the ammonia. If there is absorption of ammonia in a standard solution take place, then it will increases the color. Okay. Now the next is your procedure. Okay. So the first procedure is that we have to measure the apparent color. For that, observe the color of the sample by filling matched Nessel cylinder to the 50 ml mark with the water and compare with the standard. What we have to do, we take a 2 Nessel solution. In the first solution, we have a sample. Okay, and the next in the second Nessel cylinder, there is a standard. Now, what we have to do, we compare this both. Comparison. Compare by looking vertically downward through the cylinder towards a white surface placed at such an angle that the light is reflected upward through the column of the liquid. Okay, so when we compare this, the samples and the standard, it is compared like that. We compare it by looking it vertically downward and put the cylinder towards a white surface and place at such angle that the light are reflected upward through the column of the liquid. If there is a turbidity has not been removed, then report the color as a apparent color. Okay, because in apparent color there is no removement of the this turbidity. Okay, if the turbidity is there, then it will show the apparent color. If the color R exceeds 70 unit, then dilute the sample with the distilled water until the color is in the range of the standard. Okay. If the color of the sample are exceed to the 70 unit, then what should we do? We use our distilled water and added into it, and we are adding distilled water until the color ranges come into the range. Now the next is your measurement of the true color. Okay, so we all know that true color are measured by when we remove the turbidity. When we remove the turbidity. And this turbidity can be removed by a centrifuge. Okay, so remove the turbidity by a centrifuge the sample until the supernatant liquid is clear. Okay, so then we compare that centrifuge sample with the distilled water to ensure that the turbidity has been removed. And if the sample is clear, then it will compare with the standard. Okay. When we centrifuge it, we have a centrifuge sample. So first of all, we compare the centrifuge sample with the distilled water for the ensuring that all the turbidity are removed. And if it is clear, then we compare the sample with the standard solution. Okay, now the next is your calculation. Okay, so the color of the unit or a Hazen per unit are calculated by a 50A divided by a V. 50A divided by a V. A is what? A is the estimated color of the diluted sample, and V is the volume in ml of sample taken for a dilution. A is what? A is the estimated of the color of the diluted sample. Estimated of the color, sorry, of the estimated colors. Just a minute. And V is the volume in ml of a sample taken. Now the result. Okay, we all know that if the color of the unit or Hazen per unit are in the range of 1 to 50, then the record is nearest to a 1. And if it is the range between 51 to 100, then the record nearest 5. 101 to 250, then the record nearest 10. 250 to 500, then the record nearest 20. Okay. So this is all about the measurement of the color by using a Cobalt Method. I hope you will understand. But if you have any query related to this topic, you can ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe ITLS Academy on YouTube and get our app from a Google Play. Or you can also ask your query to the helpline number that are already mentioned in your slide. That is 7080833450, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.